Deoxyribonucleic acid, or DNA, is material that self-replicates and is always in nearly all living organisms as the carrier of genetic information. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogenous base. The phosphate and sugar groups make up what's called the backbone of DNA.
The four types of nitrogenous bases are split into two groups, purines and pyrimidines. The purines are adenine, guanine, and the pyrimidines are thymine and cytosine. And purine and pyrimidine bind together using hydrogen bonds as a complementary base pair. Adenine and thymine bind using 2 hydrogen bonds, and guanine and cytosine bind using 3 bonds.
Each of these strands has a 5' and a 3' end. The 5' is determined by the end of the deoxyribose sugar facing the end of the strand. The 3' is determined by the third point, or front face, of the deoxyribose sugar facing the end of the strand. These strands run in opposite directions, antiparallel from each other.
DNA replication is a process necessary for the growth and reproduction of an organism. Replication is a complicated process that happens constantly within replicating cells. It replicates semi-conservatively, which means that 2 copies of DNA are made from the process.
The first step in DNA replication is the making of a replication fork. It is created when DNA helicase unwinds the double helix structure of the DNA. An enzyme that helps the helicase is called a topoisomerase, which participates in the overwinding or underwinding of DNA. The replication fork looks like a fork in the road that is composed of a leading strand and a lagging strand of DNA.
An enzyme called the primicase continues the process by making a small piece of RNA called primer. The first primer is the start of a new strand of DNA.
Another enzyme called the DNA polymerase bonds the primer and begins to add nucleotides along the leading strand. The lagging strand is built in pieces due to the fact that it runs antiparallel to the leading strand in a 3' to 5' fashion. These pieces are called Okazaki fragments. They are short sequences of DNA nucleotides that are discontinuously synthesized.
A second DNA polymerase fills in the gaps between the Okazaki fragments, and once the lagging strand is filled, an enzyme called the exonuclease removes the RNA primers from both strands. Another DNA polymerase fills in the primers.
Finally, an enzyme called the DNA ligase seals the fragments of DNA in both strands to form a continuous double strand. All of the DNA polymerases have the ability to proofread the finished DNA for irregularities and correct any errors. Now that the cell has replicated with a new strand of DNA, the process can start all over again.